Hi, I'm Dana, and I will demo an analysis of a familial cancer case using the phenotype first approach. So first, let's find our case on the case dashboard. I will go to the CGAP core project that includes this publicly available breast cancer case. When I click on the case ID, I can see this case overview that demonstrates the workflow of CGAP analysis, starting with accessioning, going to bioinformatics, filtering, and then the user-facing semi-automated interpretation and case review processes. So when I start from accessioning, I can look at the accessioning report and history, as well as the patient information, family information with human phenotype ontology term on the bottom, and the full pedigree here on the right. When I click on the pedigree, I can learn that the patient is 36-year-old male with breast carcinoma, with a family history of breast carcinoma and pancreatic adenocarcinoma for the mother, prostate cancer in maternal grandfather, and ovarian neoplasm at maternal aunt. I can now upload images, I can upload documents. I need to make sure that these data do not include personal health information at this point. After reviewing my pedigree, the family history, potentially adding some notes and making changes in this interactive pedigree tool, we can go back to the case information and review the accession report and history for the proband and the additional 12 family members. As you can see in this report, only the proband was sequenced by whole genome sequencing in this case, and we can view the sample, or for simplicity, I will move forward to the bioinformatics steps of our workflow. So first thing that you can see here is the quality control dashboard that includes information about the coverage, the predicted sex that matches the submitted sex, and we have this green flag over here. We also have green flags for the heterozygosity ratio, the transition transversion ratio, and we do not see any yellow or red flags over here. So after making sure that the QC metrics looks good, we can also go to the individual files over here, to the QC, to the fast QC files, as well as the raw data for the BUM or the GVCF file. And we also can look at the provenance graph that goes all the way from the BUM file to our annotated VCF file that was ingested to the portal, and provides you with the option to learn about the bioinformatics steps, as well as linking directly to the QC and the intermediate files. At this point, after we ensure that the data looks good, we would like to do the filtering process. When I go to the next filtering step, I can see that there are already several filter blocks that were selected for breast cancer and for familial cancer. But I would just want to briefly show you how to create these filter blocks from scratch. I can add a filter block, and I can see that before filtering, we have about 4,000 variants. And after starting filtering according to position, genes, variant type, consequences, and other presets that we have here, we can go down to, for example, by selecting the breast cancer gene list, we can go down to 10 variants. So first, we will do the technical review, and this could be quickly done by clicking at the BAM snapshot. So that's similar to an IGV snapshot that shows that the variant has relatively acceptable quality in terms of the forward and reverse strand and the alternate reads that are covering this variant. So we will move this variant for the next step of interpretation. And now we would like to do the same technical review for all of the 10 variants available here. And to save us some time, I will move all of them to the interpretation step after doing the review process elsewhere. Now, when we select them to the interpretation step, you can see that the entire workflow is enabled, and we can go to the next step and review our 10 variants that we just selected. What is nice to see here is all of these variants were already classified before. So we can see that there is ACMG classification for 9 out of the 10 variants. And we can see that one variant, the MUTYH variant, was previously classified for discovery as a strong candidate in terms of the gene and weak candidate in terms of the variant. All others have ACMG classification, and interestingly enough, the BRCA2 was previously classified as a pathogenic variant. At this point, we would like to open the annotation and interpretation step, and we would like to learn more about the gene and the variant. And what we can see in the overview of the gene is information about the gene name, symbol, and gene summary from RefSeq. We can have links to some important external databases and learn more about the constraint scores. And obviously the BRCA2 is a highly constrained gene, as we can find in this dashboard. There is also an internal gene summary that was made on December 16th, 2021. And this summary summarizes what our internal knowledge base says about this gene. We can look at the variant notes, and we can, to support these notes, go to the annotation browser. And what we can find is that this variant, which is highlighted here, is an splicing variant. And there were several ClinVar submissions that show that this variant was submitted as pathogenic, and other variants at the same location were submitted as pathogenic or likely pathogenic annotations in ClinVar. So we can also see that the NOMAD database shows that there is no variation that was seen in NOMAD. And we can also see information about the orthologs over here. So based on this annotation browser and the variant annotation space that shows us the ClinVar submission, the 0 alleles in NOMAD that matches this variant, and the external resources over here, this variant was actually already noted to affect the donor splice site in intron 2 of BRCA2 gene. At this point, we are ready to do our ACMG-driven classification. So we will go to the clinical tab over here, and we will find these ACMG rules opening up. So this variant was classified as pathogenic, as you can see, and the following 3 rules were invoked. So pathogenic very strong 1 was invoked because this is a frame shift variant in a gene where loss of function is a known mechanism of disease. The pathogenic moderate 2 rule was invoked because it's absent from controls in NOMAD. And the pathogenic supporting 5 rule was invoked because the ClinVar source recently reported variant as pathogenic. So we can see here an automated calculation of pathogenic, and this calculation is following the publication of ACMG, which is could be clicked here or cited from here. We can also change our project classification rules. So sometimes labs would prefer to change how they weight those rules. And sometimes a lab can say that instead of calling this rule pathogenic very strong, they would like to call it moderate. And the moment we do that, you can see that the classification goes to uncertain significance. If we change it to strong, the classification will be likely pathogenic. Our classification here could still be pathogenic though, but what we have here is a suggestion that is automatically calculated using the ACMG calculator built in the system. So I would like to keep this as pathogenic very strong, and I would return to the case without saving the information, since it was already classified in a way that actually meets our expectations in this example. At this point, when everything was classified already, we can go to the last step of the case review, and we can assign that some of these classifications would be mentioned in our report. And specifically, I would like to make sure that the pathogenic and likely pathogenic and variants of uncertain significance would be notified as primary findings. So let's just make sure that that's happening here. At this point, I hope that you have a nice flavor of how to analyze cases using the phenotype first approach. I would like to thank you and to wish you good luck with your CGAP analysis.